Welcome back to the studio. What a session. We've had a lovely session of Spotlight Talks. Now, one of the things that we really love doing here at London Calling is bringing a platform to early career researchers. Everyone is involved in the science from PI to technician, and we really love giving a voice to some of the people earlier on the journey. So two of my colleagues here on the sofa today have just done a 15-slide and 4-minute talk to pitch. Vote for them so that you can bring back your favourite tomorrow for a longer talk. But how was it? How was it getting up there in front of 600 people?
It was fun. It was scary when they mic you up. Now you're like, oh, this is getting real, you know? But once you're on the stage, I feel like it was a little bit funny even.
Yeah, Rachel?
I was more confident on stage.
Yeah. Is that the first time you've spoken to 600 people in a room?
Yes.
On a 25-metre screen?
Yeah,
fantastic.
it was really good.
Well, look, it was such a great talk. Well done. So, actually, my first question is going to be for Kieran. Thank you for your wonderful talk. So much to take home from that. And what's really striking here at London Calling today compared to 1 year, 3 years ago is the number of people marching through the journey from research to method development to, into that translational journey. And you've spoken about the fact that you have been working on retrospective samples. You're starting to get real patients into your journey. Can you share with me your dream scenario? 1 year, 2 years from now, what does a patient journey look like when you've got nanopore sequencing helping characterize those cancers in real time? What's that look like for you?
So for sure, I think 1 year from now, I'm hoping it's just going to be integrated into, we have a cycle of sort of 2 to 4 weeks from when samples collected and it gets sequenced and gets analyzed and gets back to the tumor board. But I'm really hoping that we'll have information going live as part of that process and in part of the tumor board reports. I think some of it has already started happening. Some of the POG analysts are actually using some of the long read information, but usually because it's mostly retrospective, it's a little bit after the time. But yeah, so then things like complex SVs, I would hope that we could actually be resolving those, cross-checking tumor suppressor genes that they're actually knocked out, when we think they are, that we're not kind of, misdiagnosing somebody. Looking at methylation, can we tell that, the HRD that we see is actually real, that there's, a knocked out tumor suppressor by the promoter methylation. So I'm hoping all of that can be going into the report that we're actually giving to the oncologists at the tumor board.
Fantastic. And you obviously work in an environment where the research arm is very connected into the medical community. So are you sort of rethinking the way that tumor boards work? Is there a sort of a genome representation in the clinical meeting now? How does that practically work?
Yeah, we'll have to kind of think about, I think, and I'm not a POG analyst, so I'm not one of the people doing the presentations. My understanding is it's gone through a lot of refinements over the last kind of decade or so that it's been running, and they started off giving lots of information to the oncologists, and they've already distilled it down to some very key kind of like bite-sized points that and the oncologists have gotten familiar enough with the data that they know what to kind of ask and look for. So yeah. I think it's really just distilling it down to a few kind of key points in the report that we can feed back to them.
Interesting. Do you think the other people in the room, whether they're sort of genetic counselors, nurses, whoever it is, do you think that there is a room for them to become more familiar with these kind of technologies as well, and that become part of the process?
Absolutely, yeah. Yes, no, and medical geneticists as well. A lot of them, we've started having talks with those kind of people, and yeah. For a few of the projects we're doing, there's some other projects where we're looking at other germline disease and so forth.
Yeah.
And we're sort of figuring through what they need in terms of their own reassurances, like files, IGV sessions they can load up and explore. And
fantastic, fantastic. Well, great work. And Rachel, thank you so much for telling us about your project through the method of cricket. And actually, there were a couple of my friends who were just saying now, we were saying, well, do you think we could roll this out globally as a story where you use football in one country, cricket in another, baseball in another? The analogy must work its way around all the countries, right? And I know that you were saying to me before now, you were saying the international rollout of technologies that can help, universal problems like water quality is really important. So talk to me about your sort of international perspective and your goal for the kind of work that you're doing.
Yeah, okay. So currently my work deals with using the somatic coliphages. So they are like the small viral proxies, which can be used to determine the viral contamination. They are being recognized by the European Union directives for drinking water, and also Scottish Water has been using the somatic coliphage as indicators for water quality treatment. It is under the UCAS. So, of course, that's why we are dealing with it, that how can we implement this in the UK? Therefore, we have selected different drinking water treatment trains in order to monitor right from the source to the different interstages, and then look at how the small viral proxies can tell you what are the viral signatures that are present, which gets treated at each different step, so that the final water that you can just drink from the tap or from the bottle is safe.
Fantastic. And are you finding you're sort of starting to open up international conversations around this method development? I mean, it's a common theme across this sofa, method development that multiple people can access and use. Are you talking to people? Where in the world are you talking to people?
Oh, okay.
Or would you like to?
Yeah, I would love to talk to people about how we can develop this method to scale detection of coliphages in real time.
Yeah, fantastic. Well, talking of method development, Ruben, you took us on a whistle-stop tour of some fairly serious method development that you've been doing to elicit incredibly rich data sets. And it was amazing to see the work that you're doing to articulate using that method in understanding chemoresistance in CAR-T, for example. That was amazing. What other things could be discovered using your methodology, do you think? Or do you desire?
I think like the resistant tumor cells to CAR-T, that's like a prime interesting example of what our method can do. But our point of view was actually, let's first develop the method. Now with Nanopore, we can develop a method for which we have genome, transcriptome, epigenome at the single cell level, which is literally unseen before. And like it is with research, you work together with the lab right next to you, your neighboring labs, and they actually provide us with this interesting data, which you can let our multi-omics loose on. And I'm pretty sure when other labs or other researchers start developing this method, they can find this very interesting niche discoveries like we did with our CAR-T cells, hopefully.
Yeah, well, or hopefully not so niche. Right? Yeah.
Maybe not.
And how do you see that sort of maturing over the years? So this is sort of relatively early work in its lifetime. Do you think there's a lot of skills that need to be developed? So particularly I'm thinking about data analysis skills. How do you think about that being a pathway to expanding the use of your work?
So I think single-cell long-read RNA is getting more popular. And I think there are some pretty cool pipelines out there one can use. Single-cell long-read DNA genome is not common at all. The beauty is we actually made these pipelines ourselves. We posted on their GitHub. So everyone who wants to use our method, we got you covered. We have some pipelines out there to get you started, even for the downstream analysis. And hopefully with that, other labs can quite easily also use the multi-omics very effectively.
Fantastic.
Could I actually ask you a technical question? So you're doing genomes, right? Whole genome, single cell. What are you using for demultiplexing?
So we have our own pipeline now developed. It's on Nextflow, single cell DNA long, I think it is. Mm-hmm. And we're combining it with single cell nanoseq. Nanoseq is for RNA, DNA long is for DNA. Yeah. And our idea is to combine it in one big Nextflow pipeline. One can use whatever they want, and everything from all the pre-processing steps are in there, basically.
Yeah. I mean, I ask because like I've had some. We have a method called DLP that's kind of used by our collaborators, and they did a tech D run where they ran that on Nanopore, and it's sitting in my work backlog to try and figure out how to demultiplex it, because Dorado won't quite do that. Like it won't do the two ends. So yeah, we should connect afterwards.
We should have a talk afterwards.
Yeah.
This is the best thing when people on the sofa start talking to each other rather than talking to me. This is exactly what we're trying to achieve here, is linking people up in the community. So I guess that's my last question is, what are the conversations that you want to be having out there by the water cooler here at London Calling, or what are the big talks that you want to see over the next day and a half? Any recommendations for our online audience? Rachel, I'll go to you first.
Well, I would I would ask the audience to vote for my talk so that you can
See your
So
talk, yeah.
yeah, you can know more about how we can use this viral indicators, because these sentinels are important. We are planning to develop a real-time surveillance sensor-based method which can be used, and nanopore sequencing is just giving us the edge to use this kind of phages to track these viral enteric viruses. So.
Excellent pitch. Ruben, what about
For
you?
me, I'm more interested in more the cancer genomics point of view, and I think we already have a lot of great talks on that topic, and still to come. And for me, it's always interesting to see how we can use long reads also to transform, quote unquote, the field of cancer genomics.
Fantastic.
And so I'm having a more broad point of view on which talks I want to see, basically.
Excellent.
I want to be having a lot of conversations like this one about the demultiplexing and having a bunch of chats with Fritz about various tools he's developing that we're starting to use and will want to be using, and pick the brains of some of the Oxford Nanopore bioinformaticians about some of the software you've put out, and we have various little nuanced technical discussions we need to have about that. So.
Fantastic. Well, some of the best collaborations at London Calling are formed over the drinks, or in fact last year on the dance floor, which was also a very good source of collaboration.
It is.
Well, I got my last question is about walk-on music. Kieran, what was your walk-on music and why?
Well, we had Man Who Sold the World, and it's a little subtle dig at the fact that this is actually a corporate kind of capitalist conference, but it's invoking all these punk kind of anti-capitalist kind of vibes as part of it. So just a little dig at that.
Fair enough. An anti-punk choice. We love it. Fantastic. Well, thank you so much for coming and talking to us on the sofa today. I hope you have a lot more conversations out there in the drinks hall. Unfortunately, we're out of time on the sofa. If I can find my glasses, then I'll be able to read what's on the card here. So coming up next, you can network with your peers in the networking room online, or browse the amazing delegate posters in the poster hall. And don't forget to vote, of course, for your favourite. 2 people here to vote for on the sofa, and 1 couldn't make it to the sofa today. So also please do vote for our spotlights. Back in the auditorium, you can watch presentations from Mike Clark and also Megan Noonan on single-cell sequencing. So more on single-cell. And then following that at 10 to 4, we'll be heading into the first session in the breakout talks. Please check out the agenda for full details on that. So thanks very much indeed, and please don't forget to post using the hashtag NanoporeConf. Thank you.